Thank you. Dr. Jonathan Jenks, distinguished professor emeritus of wildlife science management, grew up on a dairy farm in Western Massachusetts. John taught and conducted research at South Dakota State University in the College of Agriculture and Biological Science and the Colleges of Wildlife and Fisheries. He retired, or maybe semi-retired is a better description, in 2021 after 29 years at South Dakota State University. Professor Jenks has conducted and broadly published research that addresses wildlife questions using ecological theory and has conducted extensive research on the ecology of large mammals of the northern Great Plains, including research of the effects of neonicotinoid insecticide exposure on white-tailed deer that was published in 2019 in Scientific Reports. Professor Jenks has published over 200 peer-reviewed papers, 9 books, and many reports and articles. He's provided leadership to the Wildlife Society and the American Society of Mammalogists at the state and national and international levels. And as Lori said today, in addition to an overview of the white-tailed deer study, Dr. Jenks will discuss research that is in the process of being published on neonic insecticides and ringneck pheasants and ongoing research in North Dakota on neonics and other mammals. I want to thank John for taking time to share his research with us. We appreciate it. So John, there's been fairly substantial research and reporting on the devastating effects of neonics on the population of pollinators and birds, particularly grassland birds. And you'll be talking about pheasants, but there doesn't seem to be much research at all on neonics and other critters. So what are these chemicals doing to us mammals?
Well, Greg, thanks for the introduction, and I'm happy to, I appreciate the invitation to talk to you about neonicotinoid insecticides on some mammals and birds of the Northern Great Plains, research that I've been involved in the recent past. I'll just go ahead and get into it. What I'm going to do is present 3 different research projects on deer, pheasants, and free-ranging mesocarnivores that I've been involved in over the past few years. So I'll just give you a little introduction on neonicotinoids to start off, just in case you're not familiar with these different chemicals, but they were created in the 1980s or so, and currently they're the most widely used insecticide in the world. They're viewed as having low toxicity to vertebrates and high toxicity to insects, although that is somewhat debatable, but they are touted for their flexibility and ease of use, and they're used in a lot of different ways. Now, this is a schematic that shows what they do with insects and potentially other species as well. You've got these molecules of a neonicotinoid insecticide, in this case, imidacloprid, that bind to these receptors and force them to stay open to overly excite the acetylcholine movement through these nerves and nerve impulses. So these species, primarily what was believed to be insects, were affected by these chemicals. But again, as was mentioned, there's very little research that's been conducted on some of these other species like deer and pheasants and so forth. So anyway, the flexibility of use, they're used in a number of different ways. Probably the top 2 would be the seed treatments that I'll talk about a little bit more later. And also this topical application in pets. We probably all want to try to keep ticks and fleas and so forth off our pets, but by doing so, at times we may be exposing them to some of these chemicals, and it could be potentially dangerous. They're used in seed dressings. In other words, the seeds are coated with the chemicals, and the reason why the chemical companies like to do this is because the seeds actually absorb the chemicals systemically, and that way the plant is protected systemically in its leaves and roots and so forth from insect damage, and the chemicals can control a broad range of insect species. There are a number of different neonics that are out there. These imidacloprid, clothianidin, thiomethoxam are probably the three most common neonics that have been used. The focus of our research was on imidacloprid and clothianidin, imidacloprid in deer and in mesocarnivores like bobcats and fisher and otters, and clothianidin in pheasants. The reason for the difference in evaluating these neonicotinoids is that some research conducted in Montana indicated that imidacloprid could be responsible for some issues with deer and elk, antler deformities, deformities in reproductive tracts of the species that had been documented by Hoy and others up in Montana. And then for relative to clothianidin, because clothianidin is primarily used on corn, we wanted to evaluate its effects on pheasants because of the close association between pheasants and corn. If you look at the use of these chemicals, it started in the 1990s, and since the 1990s, there's been increased use of neonicotinoids on a number of different agricultural crops, and a significant increase in use. If we look at the EPA maps for the US and look through from the 1990s up through about 2013, when after which it was difficult to get these types of data from the EPA, you see a significant increase in use of in this case, imidacloprid, and especially in region of eastern South Dakota and Minnesota and North Dakota, which was another big reason why we were interested in imidacloprid. If you look at clothianidin, you see the same sort of pattern of increased use from, in this case, 2004, with relatively high use, but a significantly higher use in more recent years, 2013. This was preliminary data from the EPA. Potential threats from use of from these chemicals are, because they're systemic, the chemicals end up in all parts of the crops that can be consumed by insects and also mammals and birds. One of the issues is that it's been documented that the absorption of the chemicals is only about 1 to 20%. So approximately 80% of the chemical is leached from the seeds, then for those that are these seed coatings, for example, into waterways. It ends up in surface run water due to tiling, and also potential persistence in soil with a half-life of up to, in some cases, 1,250 days. So you're talking years of these chemicals remaining in soils. Threats to wildlife for bumblebees, the LD50, where 50% of bumblebees died in these experiments, took about 4 to 5 nanograms per bee, and the exposure resulted in impaired foraging success, susceptibility to disease, as well as other issues. Work that's been done on partridges indicated that the recommended application rate resulted in death, and 20% of the application rate reduced clutch size, delayed egg laying, and there was accumulation in liver tissue. And then in rats, there have been documented morphological changes to testes and effects on hormones, thyroid hormones, for example, that result in changes in metabolic rate of the species, and also DNA. So to start with our research, we were interested in evaluating the effects of imidacloprid on white-tailed deer in a captive situation. I was had a captive herd of white-tailed deer at a facility adjacent to South Dakota State University. And our objectives were to identify imidacloprid levels in tissues of captive deer that were exposed to the chemical. In this case, based on the literature, we decided to look at liver, brain, spleen, and genital tissue in adult females. And we had different treatment levels I'll explain in a minute. We wanted to determine imidacloprid levels in these tissue levels in fawns that were born in our facility and compare those that survived versus those that died. We wanted to compare the behavior of adults and fawns exposed to imidacloprid. And then we had a set of data of tissue samples from liver and spleen that had been collected by North Dakota Game and Fish for deer that had died throughout the state from vehicle kills and grain overload and other factors that had resulted in mortalities of these animals. And the biologists with North Dakota Game and Fish collected tissue samples, and we were able to use those tissue samples to compare the results of free-ranging deer to our captive deer results. We conducted our experiments at the Wildlife Research Unit, the captive facility at South Dakota State, and we used 20 adult does, female white-tailed deer in both years. And I wanted to mention that all of our methods for pheasants and for deer were approved by the South Dakota State University Institutional Animal Care and Use Committee. We administered imidacloprid to deer through a water source just because we needed a uniform methodology or method to expose deer to the chemical. And Maine had documented imidacloprid and other neonics in wetlands in Canada. So what we did is we used concentrations from the literature to develop treatments to expose deer to. So we had a low treatment of 1,500 nanograms per liter, a moderate treatment of 3,000 nanograms per liter. And those two treatments were based on what was documented in wetlands. We, in addition, we added a high treatment of 15,000 nanograms per liter to determine an effect. And we also had a control where we just had we didn't use any imidacloprid for our control deer. So our four groups here are low level. We had 4 does, and these are the fawns that were born and the deaths for 2015 and 2016 for our low group, our moderate group, our high group, and our control group. Note that we did have fawn deaths and adult deaths in a number of our treatments, and I'll tell you after dealing, or let you know that after conducting captive research from the 1990s to 2020 or so while at SDSU. The one thing is that you can have animals that die from a number of different causes that could be related to the experimental design. It could be just random deaths that occur during the course of experiments conducted on captive animals. The data we collected were, as I mentioned, behavioral observations. We collected milk samples, blood samples for looking at, for example, thyroid hormones. We collected water consumption data, weather data, and organ samples. The focus of my presentation today is on the behavioral observations and also the organ sample data. We didn't have very many milk samples that we were able to collect. The blood samples, we did document an effect on thyroid hormones, but it was a relatively weak effect. And the water consumption data, I just wanted to mention that there was variability in that because our experiments were conducted in an open facility, so it rained during our experiments, and we documented that deer were consuming water from puddles inside the pens during the experiments. So that resulted in variation in our experimental treatments, but it also gave us an indication based on our observations that in, for example, our high-level treatment where we had 15,000 nanograms per liter, deer appeared to be avoiding consumption of the treated water to consume water in puddles or pools that developed within the pens from rain. And then I should mention that all the organ samples were from what we did is we necropsied the animals at the South Dakota State University veterinary diagnostic lab with a pathologist to ensure that we were obtaining correct samples, reasonable samples for the experiment. So we collected liver, brain, and spleen tissue, and these are the methods that we used. And up here I've got for pheasants and mesoconifers as well. We use the same laboratory methods for all our experiments, except for tissues. For example, for deer, we had all 4 tissues. For pheasants, we collected spleen and liver tissue, and for mesoconifers, we had spleen tissue, but we used the same experimental methods to determine the concentrations of the neonics in these tissue samples. And then we used a direct competitive ELISA test to actually estimate the concentration using a standard curve. And that's again for all our experiments. So this is liver concentrations, and I'm not presenting brain or genital concentrations in adult females, but this is just to give you an idea that we didn't find any difference based on our experimental design, our control low, moderate, or high for liver or brain or genital tissue. They were not significantly different from one another. And I also wanted to mention that our controls were compromised here, that we ended up having neonics show up in our control samples, and we attempted to explain what was going on here, and we suspect that through the consumption of the diet that the deer were maintained on, which included corn and soybean hulls, that there was some neonic that entered into our controls, was consumed by the controls that way as well. In addition to that, we had a cornfield adjacent to our captive facility, and it's been documented in the literature is that just dust from agricultural fields can spread neonics to adjacent areas, and we suspect that that potentially could have happened with our facility as well. So no real difference here for liver, for genital tissue, or for brain tissue. In fact, the brain-blood barrier probably reduced the amount of imidacloprid that entered the brain because our levels in brain tissue were extremely low. However, even though it wasn't significant, if you look at the values for spleen tissue, you see, and our controls were somewhat compromised. You see this increase in imidacloprid concentration as you go from control up to the high treatment level here. And it wasn't statistically significant, but when we used a different statistical analysis on this and approached significance of P, or the probability of 0.06, which was just about significant, indicating that there was an increase in a imidacloprid in spleen tissue with the increased exposure of deer to the chemical. So because of that, what we did is we used spleen then to kind of look at some of the other characteristics of a deer and the exposure of deer to imidacloprid based on concentrations in spleen tissue. For example, if we look at liver concentration in fawns that survived versus fawns that died here, we used liver. We looked at liver. We didn't find a difference. We looked at brain, and again, the levels were really low, and there was no difference between the fawns that survived versus those that died. We looked at genital tissue, and again, no significant difference for fawns that survived versus those that died. But if you look at spleen concentrations, fawns that died had 3 times the concentration of imidacloprid in spleen tissue than for fawns that survived in our captive facility there. So the indication was that there was potentially an effect of spleen concentrations related to survival of fawns in our experiments. We also looked at behavioral observations, and we looked at what we did is we observed adult females in our facility and adult fawns in our facility, and we separated those females and fawns based on spleen, what their spleen concentration of imidacloprid was, whether it was low, moderate, or high for both for the fawns and the adults in the experiment. And it turns out that there were some significant differences across the number of different behavioral observations that we collected. For example, walking, standing, running, laying down, eating, and others, standing and ruminating, laying and ruminating, that sort of thing. And we documented that for adult females with that had high levels of imidacloprid in spleen tissue, we had a reduced amount of foraging or eating, and a reduced amount an increased amount of inactivity or laying in the facility. And we noticed a similar thing with fawns, that there was increased amount of laying or inactivity for fawns in the facility that had high levels of imidacloprid in spleen tissue. So our thoughts there were that these adults and fawns with lower activity likely had increased susceptibility, especially the fawns, to predation, but also the thriftiness of the females could have been compromised by high levels or exposure to high levels of imidacloprid. So the next thing we did is, as I mentioned, we had these approximately 350 tissue samples for liver and spleen from free-ranging deer in North Dakota, and we wanted to compare those levels of imidacloprid in those tissues to those of our captive deer. So I've got this somewhat highlighted here, but if we look at the mean levels here, and we compared, for example, liver concentrations for captive deer versus free-ranging deer in North Dakota, we see that free-ranging deer had about 3 times the concentration of imidacloprid in spleen tissue than captive deer in our facility. And also for the spleen tissue, relative to captive deer versus free-ranging deer in North Dakota, the free-ranging deer had about 3 times the level of imidacloprid in spleen tissue than the captive deer in our experiments at South Dakota State University. So the implications for free-ranging deer in North Dakota, and based on our experimental work at South Dakota State University are that deer exposure in North Dakota averaged 52% for the samples that were collected from 2009 to 2017. In other words, 52% of approximately 352 50 samples, 52% showed were exposed to imidacloprid, had imidacloprid concentrations in the tissue. The imidacloprid in North Dakota deer spleens increased by an average of 0.11 nanograms per gram per year from 2009 to 2017. So the indication was there was increased exposure to deer in North Dakota over that time series. And then finally, 77% of North Dakota deer that were exposed to the imidacloprid or that had imidacloprid in spleens or had levels in spleens equal to or above 0.33 nanograms per gram, which was the mean level in spleens of captive fawns that died in the experiments in our captive facility. So that essentially, just to further explain this, of these 52% of deer that had imidacloprid, 77% had high levels or levels higher than fawns that died in the facility. So this is the end of the experiments on white-tailed deer. And now I'm going to jump right into the experiments we did with ring-decked pheasants, which was sort of a companion project to the deer project. Both of these projects were funded by South Dakota Department of Game, Fish, and Parks, and they both, biologists from the state were concerned about deer, but they were also concerned about pheasants because pheasants are, of course, one of the #1 species in wildlife species in South Dakota, and because 30 to 40% of the diet consists of agricultural seeds, and there had been documented observations of pheasants consuming seeds in springtime, post-planting, and also seedlings in April and June. So there was a concern there that there was significant exposure to treated seeds from pheasants in the state. So our objectives were to determine seed selection because treated seeds are dyed. So we wanted to look at if pheasants were avoiding seeds that were treated with neonics, and in this case, as I had mentioned, we were concerned about clothianidin on pheasants. So we looked at determining if exposure to this neonic had an effect on survival, and we also investigated clothianidin effects on breeding. We looked at recruitment effects and chick health in our experiments. We conducted these experiments at the South Dakota State Fairground Poultry Building in Huron, South Dakota, in spring of 2016 and 2017 for the survival experiments, and in 2017 for the breeding experiments. The first experiment was conducted on seed selection. So what we did is we observed captive pheasants feeding on seeds and determined that they consumed approximately an average of 75 seeds, and that comes into play later. But what we did is we exposed them to treated, this should be treated and dyed seeds that were treated with clothianidin. Dyed seeds, they weren't treated with clothianidin, but they were dyed similar to the treated and dyed seeds, and then untreated seeds that were undyed. And then we looked at, we provided those seeds in equal availabilities to pheasants in control experiments. We had 8 groups of pheasants that were that we used in our trials, and then we looked at the seeds that were consumed. And in 7 of the trials, the pheasants selected for undyed, untreated seeds. In one of the trials, the pheasants selected for treated dyed seeds, so about 12% of the pheasants then in our trial selected for the treated seeds, but the the 88% of the pheasants avoided treated and dyed seeds. So the indication, sort of similar to what I kind of mentioned with deer, we had observations that sort of indicated that deer were avoiding a consumption of treated water, and we suspected that maybe that's because of some sort of astringency associated with the chemical in the water that caused or a secondary effect that caused the deer to avoid consumption of the water when they potentially could. We saw a similar thing with pheasants avoiding consumption of treated seeds. For survival methods, as I mentioned, we observed pheasants just consuming seeds, and we documented that they consumed on average about 75 seeds per day. And so because of that, we based our experimental design on that 75 seeds. We orally administered seeds during our trials over a 30-day period and monitored 60 days post-treatment and looked at weights and euthanized and necropsied the pheasants. And again, all of these experiments were approved by the Institutional Animal Care and Use Committee at South Dakota State University. But you can see our treatments here were the amount of treated seeds then. We had a control with no treated seeds presented to the captive birds. So in this case, we fed them 75 untreated seeds. And then in our low treatment, low concentrations or low treatments, we had 1 treated seed and 74 untreated seeds, 2 treated seeds and 73 untreated seeds, and then 15 treated seeds. And then finally, all the seeds that were fed, 75 were treated. So that would have been our high treatment level then. And you can see the different milligrams of clothianidin and how they increased with treatment for our experimental trials. We had 5 males and 10 females per treatment level, and used the same administrative protocols over a 14-day period. For our breeding methods, where we were looking at allowing the birds to breed and then looking at characteristics of eggs and also survival of birds as well. We placed them together and allowed them to breed with 1 male and 2 females, and then collected the eggs, looked at egg shell thickness, width, length, volume, and placed them in an incubator, and allowed them to hatch, and viewed them every 5 days. And then once the eggs hatched, we measured the chicks, looked at unhatched eggs, opened them, and looked at characteristics of the unhatched eggs as well, based on days of development. The chicks were measured on day 8, 16, and 32 after initial measurements, and compared. And first of all, for the survival, we had some variability in our survival, but the take-home message here is there was approximately a 20% decline in survival with 75 with consumption of the females having, where we fed them 75 treated seeds, when we compared those to like 15 treated seeds, or those females, 2, 1, and 0 treated seeds. And we did have our controls were somewhat compromised as well, because we did document that there was some clothianidin in the seeds that we fed to even our control birds. For the breeding results, if we look at 75 treated seeds here, the heavy treatment or high treatment versus our control treatment down at the bottom here, there's a lot of variability, but for the most part, we had a hatch percentage per group of about 20%, Number of chicks was low compared to our control. Chick survival was about 20% reduced compared to other treatments, or an average of other treatments in our experimental design. And there was a reduced, also, if you kind of look at the number of chicks, I mentioned that, but the number of chicks for our control treatment at 38 chicks born versus, or hatched versus 16 number of chicks in our 75 treated seeds treatment. So lower number of chicks born. Now, in addition to the captive work on pheasants, we also went into the field. Actually, we didn't, but the state went into the field and collected free-ranging hen pheasants in April during the year throughout eastern South Dakota. And this just shows you where the pheasants were collected. And what we did is we collected those birds and we necropsied them, and we collected liver and spleen concentrations. But again, we didn't have spleens for all birds in the sample. We had about 180 birds that were collected total. And this graphic is to show you that the concentrations in liver and spleen were very close to one another. And so what we did for pheasants was focused on the clothianidin concentration in liver samples to increase our sample size, and also because based on the literature, other research that had been conducted on partridge had looked at liver concentrations of neonics. So that was the rationale behind using liver concentrations instead of spleen concentrations. We look at average ELISA values, nanograms per gram, in each of the regions of South Dakota. We documented exposure in all of the regions with, and they weren't significantly different from one another, but the southeast region of South Dakota had tended to higher levels, average levels, with 3.3 nanograms per gram in liver samples, versus the northeast and south central regions having somewhat lower levels, average levels in liver samples in female pheasants. However, we did notice some birds that had high levels. Now, we are talking about 2 to 3 nanograms per gram, but there were birds, approximately 4% of the sample, that had relatively high levels, greater than, in some instances, greater than 12 nanograms per gram in liver samples, indicating high exposure. And if I go back to the white-tailed deer experiments, some deer in North Dakota had spleen concentrations of greater than 8 nanograms per gram in spleen tissue, compared to the average levels of, as I mentioned, like approximately 1.5 nanograms per gram in liver tissue, and less than that in spleen tissue. The initial nesting period, I should mention that, well, I sort of mentioned that other egg characteristics that we measured were not significantly different between treatments. However, initial nesting period was delayed about a week for pheasants consuming the 75 treated seeds from about 29 April to the 5th May. So about a week or so delay in nesting for those birds compared to birds in the control treatment. So the implications for free-ranging pheasants are that pheasants avoided consuming treated seeds. Adult and chick survival was reduced about 20%, and about 12% of captive pheasants selected for treated seeds, and about 4% of the wild birds had really high, relatively high concentrations of clothianidin in liver samples. And now I think I better push through the mesocarnivore data. For North Dakota here, we looked at bobcats and fisher and otter. We just had a couple of objectives for these mesoconifers. We just wanted to assess exposure in the 3 species based on imidacloprid using spleen samples, and we also wanted to determine if otters would have higher concentrations of imidacloprid than more terrestrial species due to the aquatic nature of otters. North Dakota collected spleen tissue samples from harvested animals throughout the state, but note that the bobcats were collected in western North Dakota, and fisher and otter samples were primarily collected in the eastern region of the state. This just shows you the age and sex distribution of the sample. We had about 100 samples for bobcat, fisher, and otter, and approximately equal numbers of females and males that were collected for evaluation of the imidacloprid in these spleen tissue samples. So we were able to look at sex, we were able to look at age, and look at actually where the samples came from as far as the regions of the state. If we look at the concentrations, they really weren't that different from one another, and otters weren't higher than a fisher and bobcat, but relative to, if you think of pheasants with 2 nanograms per gram and deer in our captive facility of 0.3, and fawns that died of 0.3 nanograms per gram, you've got 3 to 4 nanograms per gram in these 3 species. And note the prevalence too, which was sort of interesting. 13% prevalence of the 100 samples of bobcat, 15 of fisher, and 35% of otter. So there was an increase in prevalence in otter over the fisher and bobcat, likely because of their association with water is what we kind of documented there. And also note that we had 52% prevalence in deer in our sample for the free-ranging deer in North Dakota. For our analyses, we just conducted some simple models for age, Julian date, distance to water for the 3 species. And really the only thing that was significant was Julian date and distance to water for otters, and those variables were not significantly different for the other species. For the Julian date, we documented that really what happened is that peak exposure for river otters appeared to be about the 1st of July, but there was a lot of variation in these data. So there's, so even though the peak appears to be around the 1st of July, there is variation there. And note that for other species like free-ranging deer in North Dakota, there was no effect of time period when the sample was collected. For sex and major watersheds for spleens of otter, fisher, and bobcats, the only significant finding was minor watersheds with river otters. In other words, there were differences in concentrations of imidacloprid in spleens of river otters in different minor watersheds, but no differences in these other species that aren't really tied to water sources. So if we look at the results here for example, the Forest River, the Park River, which had the highest concentration of imidacloprid in spleens of river otters, Cheyenne River, and the Wild Rice River, those were the higher levels, but there were some rivers where mean concentrations were actually 0 there. So there was quite a bit of there in our spleens of the species of otters that were collected in those systems. So there was quite a bit of variation, which is, when you think of 35% or so prevalence, that's why you see the variation in concentration levels that are associated with these minor water systems within the state. So the implications then for mesoconifers are that we did detect imidacloprid in spleens of the 3 species in North Dakota, that the imidacloprid concentration in spleens of otters varied by minor watershed. We did see an increase in prevalence of exposure of otters compared to fisher and bobcats relative to imidacloprid and spleen tissues as well. So with that, I think I'm okay. I wanted to acknowledge the South Dakota Agricultural Experiment Station, Federal Aid to Wildlife Restoration, and the South Dakota State University Department of Natural Resource Management that supported and funded the research that was conducted in the studies that I just presented to you. And with that, I'm happy to answer any questions you might have on the research that I've been involved in. Thanks again for inviting me to talk on these projects.
Thank you, Dr. Jenks. With that, we're going to invite folks to ask questions. Can you make the assumption that in the wild, there is a specific source for, say, otters, pheasants, and deer specifically that neonics are being introduced? It seems like water for otters, seed for birds, and maybe forage for deer. Is that an assumption?
Well, as well as water for deer, if they have to consume water, and if they're consuming water out of wetlands that are contaminated with neonics, that could potentially be how deer are being exposed to the chemicals. But you're correct in that forage is another way that consumption of forage could be another way that these deer or agricultural crops where they're being exposed to the neonics. For otters, we suspect that consumption of fish too, although we did not look at concentrations of fish within these stream systems in North Dakota because otters are tied to consumption of fish. That is a potential for how these otter are being exposed to the chemicals. And for pheasants, yes, treated seeds as well as agricultural crops are potentially how pheasants are becoming exposed to the neonics, as well as water sources too.
Right. So if you have a question, please use the raise hand tool. There's, okay. Ask to Greg, you want to go ahead and ask your question?
Yeah, thank you. So I'm Greg Stanley, I'm a reporter over at the Minneapolis Star Tribune. And so it just looks like, so it looks like you guys were able to find, so there is a difference between the high concentrations of both deer and pheasant and survival rates with the spleen concentrations. And just given the prevalence you're able to find too on how many of them are in the wild have those high concentrations. Are you right? Can you say, are these neonics, are they killing wild deer, wild pheasants, or reducing survival?
It's a really good question. It would be nice to and we weren't able to do this, but I was involved with some free-ranging deer studies in North Dakota, where we radio collared fawns of deer and looked at fawn survival. However, at the time, I wasn't involved in the neonic research, so we did not document neonic levels in fawns that did die in North Dakota. And of course, we wouldn't want to kill pheasant deer fawns that didn't die in order to document neonic concentrations in them, because our objective in those studies was to document survival of fawns. So unfortunately, we don't have any data that does tie survival in free-ranging deer and pheasants directly to consumption of these neonics. And that is work that needs to be done.
Thank you, Dr. Jenks. Representative Hansen, do you want to go ahead and unmute and ask your question?
Thank you, Lori and Dr. Jenks. Very Very informative presentation. I have a question and then a comment, or a comment and question. We, for 2 years, the Minnesota Department of Natural Resources has collected spleens of deer to look at neonicotinoids. The first was a very broad collection, and the second year was a very limited, and that became very limited because of intensive engagement from the agricultural industry. And I don't know if they're going to do the neonicotinoid study in spleens this year, and I don't believe any results have been published. So one, my question is, have you engaged with them at all on their research? And then the second question is, have you felt any pressure, intimidation, or threats from the agricultural industry, whether it's the agricultural chemical industry or the commodity or farm groups?
Okay, the first is yes. One of my co-authors is Eric Mitchell, who works for Minnesota DNR, is involved in work that's being conducted in Minnesota. And I have been interacting with Minnesota DNR biologists with work that's ongoing on imidacloprid deer in Minnesota. So I have been involved in working with them. The second question, yeah, the chemical companies did actually, they were not happy with what we documented in, for example, deer. They questioned our results. They told us that they went out somewhere in Missouri around, I think it was St. Louis, and collected 10 deer, and they didn't document any neonics in them, so we couldn't have documented any neonics in deer in the Dakotas. And they questioned our methods using the ELISA test for documenting exposure to neonic. However, as I had mentioned during my presentation, we use the same methods for all of our experiments, and we have been reviewed by the British Chemical Society, and the folks that reviewed us had said, yeah, it would have been nice if we used a better methodology like gas liquid chromatography rather than colorimetric chromatography for our laboratory experiments. However, they acknowledged that the study was of significance. And then secondly, the work that has been done after our work that has looked at gas liquid chromatography has documented that there has increased exposure to these neonics over what we documented with the colorimetric methodology. So in other words, our methods are more conservative than what actually likely is occurring in the wild. But I've experienced, we've experienced some quite a bit of pushback from the chemical companies.
I could follow up.
Go ahead, Rick.
Yeah, thank you, Lori. So Dr. Jenks, just this is published, peer-reviewed, and there is other work occurring around the country or around the world on similar things for reproducibility or similar research. Is that correct?
Okay. Well, there is other ongoing work on different species. The deer work has been published in scientific reports. The pheasant work, I'm behind on that, but I'm responsible for getting that submitted and published. The mesocarnivore work has been submitted to the Journal of Wildlife, Journal of Mammalogy, and is being reviewed right now.
Greg Larson, did you have a question? Go ahead, please.
Yeah, I was just wondering, Dr. Jenks, what questions do you still have? If money and resources weren't an issue and you could just do 1 or 2 research projects around this, what would you do? What questions are still in your mind that you think are important that need to be answered?
Okay, one of the questions that has consistently come up that we've dealt with or tried to deal with is, okay, well, if these neonics are water soluble, there's been some controversy about, okay, well, where are they stored in the body then? Are they stored in fat tissue? Are they stored in the spleen? Where are they stored? And we don't know where that is. So more work needs to be done to find out where the chemicals are being stored, how they're being stored, and do they stay in the animal long term, or are they? Because when you think of a wild animal, it's not like a human that we store fat, and it's difficult for us to get rid of that fat, at times, unless we go out and get into some exercise program. These animals actually use this fat throughout the year during the winter to survive winter and so forth. So you get the animals using the fat. So is the chemical being stored in the fat or someplace else in the body? So that ends up being important to find out. Also, exposure levels. We documented a lot of variability in exposure. And as the previous question had mentioned, how are they getting how are they getting the chemical? Why are some deer why do some deer have 0 concentrations of imidacloprid in spleens versus other deer that have high levels of exposure? Is it due to variation in the environment? Is it due to behavior in some way and how they're interacting with their environment? Are they avoiding? Are some deer avoiding exposure to the chemicals? And I think more work needs to be done on that, both for pheasants and deer, as well as the mesocarnivores. So that would be another avenue of research that needs to be conducted. As far as I'm going to respond to the research in other regions of the country too. I should mention that in personal conversations with researchers in other universities in the country, a number of them have stated that because of the potential interaction with agriculture, they have been unable to or not interested in getting into research in this area because of negative the potential for negative feedback on research there. So I should tell you that, and there's a lot of, as I mentioned, pushback from the chemical companies and potentially at universities as well. I hope I answered your question.
Thank you. John Bly, you have a question. Please go ahead and unmute yourself.
Thank you. And thank you, Dr. Jenks, for sharing this with all of us. My question is in a couple parts. First, just am I accurate in recalling that the mesocarnivores that were exposed had higher levels of neonics in their spleens?
Their average concentrations based on the methods that we used, although we did not statistically compare them to the pheasants and deer, the average levels appear to be higher in the mesocarnivores than the average levels in the other species. Okay.
And do you think that's a result of like bioaccumulation?
Potentially. That's a very good comment. It's potentially that could be the case.
Okay. And so my last question is, do you have any idea on how neonics could be bioaccumulating in humans?
Very good. A very good question. Of course, I don't conduct research on humans. I can't say anything there. But there is concern about neonics in water systems that in human exposure to these neonics that has been documented in literature and in agricultural crops as well. There was a study that was done that found neonics in foods in the congressional cafeteria in Washington, D.C., indicating potential exposure to our senators and representatives in Washington, D.C., for example. And there have been other studies as well in Japan, for example, that have documented high levels of neonics in infants in Japan, and there have been other studies as well. There was a letter that was written to the director of the EPA concerned, I think it was the Defenders of Wildlife, but I'm not absolutely positive on that, but it was a letter written to the director of the EPA with a concern on exposure of pregnant women and infants to neonics due to the seed dressings, and the fact that a lot of the chemical in the seed dressings was ending up in water systems and potentially exposing pregnant women and infants to these chemicals. So yeah, there's concern.
Thank you for that good question, John. Jared, do you want to go ahead and ask your question?
Sure. Thanks, Dr. Jenks, for the presentation. I found it pretty interesting. A comment and then a question. You know, the treated seed debate is interesting to me as a guy that, you know, manages for upland birds on my own property. You know, when I planted a food plot this year, I had somebody else come in and do it, and I'm not sure if they had the wrong plates in or what, but they dropped a lot of treated seed on the landscape. And shortly after that, you know, I see roosters and hens out there doing their courtship stuff, but they're also out there foraging, and I'm going out there clapping my hands trying to get them out of there because I'm worried about you know, what's going to happen when they're eating that treated seed. And I got it fixed and tilled under after that. But my main question would be, if you go back to there's one particular slide that I had a question on, and it would be the under the breeding results slide. Could you tease out those? We covered it briefly, but could you tease out those yellow numbers one more time and what you think that means as far as implications go for pheasants?
I brought it up here now. Can everyone see this?
Yeah.
Okay, well, I'm just the chick survival is that just generally we document about a 20% decrease in chick survival with high exposure. The 75 treated seeds consumed by the hens resulted in about a 20% decline in chick survival for that treatment. And also, if you look at the number of chicks, if you look at even though the 15 seeds is pretty low number of chicks too that were hatched, if you look at these low treatments in the control, you had higher levels of chicks that were actually hatched for the most part versus the higher treatment levels here. And the hatch percentage per group here, note that it was at the lowest level of about 19% per group for the 75 treated seeds. Was low for the one treated seeds too, but as I mentioned, even for deer, there's a lot of variation in the data, especially when you get into captive research here. But I think that it's safe to say that the hatch percentage was low, chick survival was lower, number of chicks hatched was lower for the high clothianidin treatment of 75 treated seeds per adult hen. Does that help?
Yes, sir. Thanks for teasing that out for me.
Greg Larson, do you want to go ahead and ask your question? And then Donnie, you're next.
Yeah, I was just, I think I saw in one of your slides, John, that that there seemed to be increasing amounts of neonics in spleens over time. Is that correct? That there was lesser amounts in earlier years and more in later years. Was I correct in that?
Yes, this is the slide here.
Mhm.
Yeah, right here, the second point here, that I did a regression analysis, which was significant, and it was indicated that concentration in the spleens of free-ranging deer in North Dakota was increased by about average of 0.11 nanograms per gram per year from 2009 to 2017, which sort of indicated that either these neonics are increasing in the environment, and exposure to the neonic is increasing in deer as due to increase of the neonics in the environment. We did not see a relationship with age, so it appears that it's more likely in the environment that because of the half-life of these neonics could be, for example, 1,200 days or a few years, and the farmers are using these every year, it could result in increased amount of the chemical in the environment through time. Is that what you're referring to?
Yeah, that was good. I mean, you know, in the Midwest, where I think 95% of corn is treated seeds and more than 50% of soybeans is treated seeds. And in a rotation of corn, soybeans, where, you know, in Minnesota, there's like 15,000 acres of corn and soybeans planted, that there isn't a year between that the amount of concentration would be going down. So if I got a half-life of up to 1,000 days, it would seem to me that the levels somewhere in the water, in the soil somewhere, is you know, rapidly increasing. So do you think that would, so you're saying that might be the reason for the increase in the concentration in the spleens over that period? Is that
correct? Potentially, that's where it's coming from. Of course, we didn't have background levels of neonic over the time period of this 2009-2017, so we can't say for sure, but that is the potential for where that's coming from. Sure.
Thank you.
Thank you, Dr. Jenks and Greg, for your question. Bonnie, can you go with yours?
Thank you. I'm thinking about the European Union having banned neonics, not based on a lot of research, but because of a precautionary, just lost it. But out of an abundance of caution, before more research is done, banning neonics, are you seeing any kind of movement in either North Dakota and or South Dakota's Department of Agriculture, Game, Fish, and Parks, or your legislators in terms of policy change or addressing this issue at all?
Well, in North Dakota, because we had biologists on this study with North Dakota Game and Fish that we collaborated with, they brought this information directly to their habitat biologists, and they changed their policy in North Dakota so they do not use treated seeds on any of their managed lands anymore. That has not been the case in South Dakota, to my knowledge. But that, you know, that is probably the only thing. I do not have any information on policy related to the Department of Agriculture in these states or legislatures or anything like that.
Well, at least that's one step in the right direction. Thank you.
Well, I'm going to ask for any other questions at this time. It looks like, Greg, you have a question, and then Any others, please raise your hand because we're nearing the end.
There was, yeah, John, there was a study I read, I think coming from Canada, that showed that in, I think it was white-throated sparrows, that that it didn't appear that, at least from that study, that actually killed the bird, but it reduced the viability as far as foraging and the delay in migration and that. And I was wondering if did you see any of that in the pheasant study?
We did see some activity. I didn't refresh my memory on those results, but there was, we did see some activity in the captive birds. But what I found through my career is that if you really want to document that kind of effect, you really need to go in the wild and do that. And because for the pheasant study that we did, all that, the free-ranging part of both deer and pheasants, we're just collecting samples and then looking at concentrations in those samples. We weren't able to actually document that occurring. But if you recall, when I presented the deer information where we looked at the behavior, we did document reduced feeding and reduced activity of fawns and adults. So we suspect that that might translate into reduced feeding and activity of deer in the wild. And if so, it might make the animals, especially the fawns, more susceptible to predation. Of course, we did not document that. So it's circumstantial at best right now.
Thank you, Dr. Jenks. So final call for questions. In the meantime, Dr. Jenks, what is the most disturbing finding, and what would you hope to come out of this research? What change is needed?
I think that that we need to evaluate wise use of these chemicals to make sure they're not having a significant impact on wildlife populations. I should mention that we did document, like in North Dakota along the Red River, we had an adult deer that died of stomach cancer, for example. When you think of a deer dying of stomach, you don't think of deer dying of stomach cancer, for example. We had fawns that were born in North Dakota that could not urinate, and in South Dakota actually, that could not urinate and died within 48 hours because of potential effects of a chemical exposure, although not documented, on reproductive tracts or internal anatomy of these animals. And so if there is an association between use of these chemicals and high exposure in deer, I think that needs to be evaluated further, and so that we minimize our effects on wildlife.